Evidence of metabolic defects demonstrated that genes specify proteins and create phenotypes. However, there are many different ideas for what a gene really is. One of the earliest is coming from the experiment that you can see here, which was the Beadle and Tatum experiment, and they were able to establish the link between genes and enzymes by exploring metabolism of bread mold. They bombarded it with X-rays and they looked at the survivors, and they determined eventually that there was strong evidence for what they called one gene, one protein. Basically, that one gene was equal to one enzyme, and all proteins are coded by genes, but not all proteins are enzymes, so therefore one gene is equal to one protein. Okay, we know now that each protein has its own gene. However, many proteins are composed of several polypeptides. So is it one gene, one enzyme, one gene, one protein, or now one gene, one polypeptide? Well, many genes only code for something like RNA, and many genes can code for many products. So it gets really complicated. Overall, defining a gene is problematic, like we said, because one gene can code for several protein products, some gene codes only for RNA, two genes can overlap, and there are many other complications. So the best definition we have is that a gene is a specific location on a chromosome consisting of a segment of DNA that codes for a particular protein or RNA molecule that has a function in an organism. That is a complicated definition. Genes are cellular units. They determine how an organism inherits characters, and the particular protein or RNA or whatever determines how those characteristics will be expressed within the organism. While defining a gene may be problematic, we do know that genes provide the instructions for making specific proteins. The bridge between DNA and protein synthesis is the nucleic acid RNA, and it's chemically similar to DNA, except of small differences that we are aware. And within those structures, we see that there is this triplet code that exists. Okay, the triplet code, there are 3 consecutive bases that specify an amino acid, creating 4 to the 3rd possible code words. The genetic instructions are written in DNA as a series of non-overlapping 3 nucleotide words, essentially, that can be translated into amino acid sentences. 61 of the 64 triplets code for amino acids. AUG not only codes for the amino acid methionine, but also indicates the start or initiation. So it's going to put a methionine wherever AUG is, but a protein will always start with AUG methionine. And there are several codons that may specify the same amino acid, so there is redundancy to the code, but no codon specifies more than one amino acid. So while there is redundancy to the code, there is no ambiguity here. The redundancy is not random. In many cases, you'll notice that it is the 3rd nucleotide of the triplet that can be changed, and that is called wobble. So there is a looseness to the binding, where the 3rd one doesn't matter so much. Now, there is a near universality for this genetic code, so it's clear that all language here is shared by living organisms because it evolved from early history. But some of the amino acids have changed what they code for in other organisms. Some of the stop codons, for example, code for an amino acid and not a stop, so there are some exceptions. But overall, the universality is a good indicator that this process comes from common ancestry. Information content of genes is in the form of specific sequences of nucleotides along the DNA strands inherited that then leads to specific traits by dictating the synthesis of proteins and RNA. Gene expression is the process by which DNA directs protein synthesis, and it includes 2 stages, transcription and translation. In a bacterial cell, transcription and translation occur at the same place at the same time because there is no separation, there is no nucleus to control that process. So it is a simpler process that occurs faster and will have less control points. The eukaryotic process is significantly more complicated. There are multiple steps to this process because of the nucleus, so it's spatially and temporally separated. The DNA will produce a pre-mRNA. That pre-mRNA will be modified by RNA processing into the final functional mRNA. That mRNA will then leave the nucleus where ribosomes will turn it into a protein and translation will occur. So we see that the process, while related and similar due to common ancestry, has changed significantly due to compartmentalization. Overall, the general stages of transcription, though, are pretty straightforward. To extract a message from the genetic code, it requires a specific starting point, which establishes the reading frame. RNA polymerase is going to bind to a promoter sequence on DNA upstream or in front of the targeted gene. That RNA polymerase will separate the DNA strands at the appropriate point and join RNA nucleotides complementary to the DNA template. Like DNA polymerases, RNA polymerases can only assemble in a 5-3, but unlike DNA polymerases, they are able to start from scratch. They don't need a primer because they are designed typically to be single-stranded within that. So it's going to attach to the promoter and will continue this process. In prokaryotes, that's pretty much it for the process. The RNA polymerase will bind and will produce and go. It gets a little more complicated in eukaryotes, as we'll see in just a moment. Overall, though, we do need to realize that DNA is double-stranded, so there's going to be a template strand. That's going to be the strand that is being copied, and then you're going to have the opposite strand, which is the coding strand. It's called the coding strand because it's going to have the same sequence as the mRNA, but will use thymine instead of uracil. The mechanisms of termination will also be different, and we'll get to that as we go over on. But overall, we realize that the direction of transcription is going to be going into this process, and it all depends upon where the reading frame starts. In eukaryotes, it gets a little more complicated. Transcription factors are other accessory molecules that will aid in the binding of RNA polymerase. That is something that is different. The transcription initiation complex is when all of these things bind together. A crucial promoter sequence that is for eukaryotes is the so-called TATA box, TATAAAA. That is indicative of the more common binding space. And RNA polymerases, there are multiple RNA polymerases in eukaryotic transcription. The one that you need to be aware of is RNA polymerase II, because RNA polymerase II is the one that makes mRNA, which is what we're looking at the most right now. But other RNA polymerases will be used to produce different RNAs. Once the appropriate transcription factors are attached to the promoter DNA and the polymerase is bound in the correct orientation, the enzymes will unwind the strand and will begin to produce this process. It's going to do about 10 to 20 bases at a time, adding it to the 3' end, growing along. A single gene can be transcribed simultaneously by several RNA polymerases at a time, but it's going to be producing this pre-mRNA, this primary production that will then be led into RNA processing. In RNA processing, your final RNA is more complicated than just an RNA sequence, and we can see the final RNA here. RNA processing is going to require changes to the process, to the actual structures. The first thing that's going to happen is that the mRNA that is being produced will have a cap and a tail added to it. The 5' GTP cap is a protective cap that will also serve as an energy source for the binding to the ribosomes. The poly A tail is again protective, preventing digestion of useful mRNA. You will also notice that the start codon and stop codon are not at the beginning of the mRNAs. So the start and stops are just where the mRNA is. There are other components in what's called a UTR, or untranslated region. These are regulatory and serve a variety of processes, some of which we are not aware of. We used to think they were junk DNA, but it clearly isn't. And then the third step is that the DNA will be processed so that the introns are removed and the exons are expressed. Introns are non-coding mRNA processes that can be altered through a process called RNA splicing. A small RNA, called the small nuclear RNA, those are going to come in and bind to this process and produce spliceosomes, which is a complex of proteins and RNAs that splice these together. The introns will splice, be cut, digested, and removed, and the exons will be attached. You can change the pattern of attachment, making different exon structures, which allows for flexibility. This is why the eukaryotic proteome, the total proteins that we can produce, is greater than our genome, which is not true for bacteria. Different types of RNA. There's more than 30 that have been described at this point, and more that we are discovering. The mRNA obviously is going to be the one that connects DNA to proteins. tRNA is going to bring amino acids to the ribosomes, and the ribosomes are composed of rRNA and proteins. There are many other RNAs, like the small nuclear RNA that we just looked at, that form part of the spliceosome structure. Okay, and then something like the miRNA, for example, the microRNA that are involved in regulation, which we'll look at in a later unit. So overall, we need to realize that RNA is incredibly important. Okay, not only is RNA important because it has so many jobs, but it also needs to realize that RNA is catalytic. So RNA world hypothesis, if you remember, that ribozyme capability, the discovery of ribosomes rendered obsolete the idea that all biological catalysts are proteins and formed one of the major foundations for the theories of the origin of life. Of translation, a cell translates a genetic message to build a polypeptide. tRNAs, or transfer RNAs, are molecular bridges that connect the mRNA codon to the amino acids they encode. One end of each tRNA has a sequence that is the opposite of the codon, the anticodon, which will allow it to bind to specific mRNAs. The interpreter, then, is tRNA. It is going to be attached by enzymes that we'll look at in a moment, but the tRNA, rRNA copy or comparison there, that's all working together. So M, T, and rRNA work together to produce proteins in this structure. Acid is joined to the correct tRNA by amino acyl tRNA synthetase. Sorry, that's a hard word to say. The 20 different synthetases match the 20 different amino acids, and each has active sites for only a specific tRNA amino acid combination. There is, however, some versatility, this correct match with that third base, that wobble. There's flexibility in the base pairing, and wobble explains why the synonymous codons for a given amino acid most often differ in their third base, not in their other bases. So ATP is used, it's enzymatic process. The tRNA will then transfer this amino acid from the environment of the cell to the actual ribosome. You have three binding sites within the ribosome, the A, the P, and the E. The A site is the amino acyl tRNA binding site, so that is where the amino acids will enter. You have the P site where the peptidyl bond will be made, the protein will be made, and the protein will form moving out of that exit tunnel. And then you have the E site where the tRNA will be able to leave the cell. So the ribosome holds the tRNA and the mRNA in close proximity, allowing for the addition to the carboxyl ends of the growing polypeptide, catalyzing the formation of the peptide bond. Evidence supports the hypothesis that rRNA, not protein, is responsible for the ribosome structure and functions. Proteins on the exterior support the shape changes of the rRNA molecules as they carry out catalysis, but rRNA is the main constituent at the interface between the two subunits. So basically, a ribosome can be regarded as one massive ribozyme structure. Can be divided into three stages: initiation, elongation, and termination. All three stages require protein factors that aid the process. Both initiation and elongation require energy that is provided from GTP. Notice GTP, not ATP. Minor difference, but difference nonetheless. Initiation is going to bring together mRNA, a tRNA with the first amino acid, which is methionine, and the two ribosomal subunits. First, a small subunit will bind to the mRNA and a special initiator tRNA, which carries methionine. In bacteria, the binding occurs at a specific RNA sequence just upstream of the start codon. In eukaryotes, the small subunit with the initial tRNA is already bound, binding to the 5' cap of the mRNA, and then moves downstream along the mRNA until it reaches the start codon, which will then initiate the binding of the large subunit. Energy in the form of GTP is invested to form this complex. Elongation stages, amino acids are added one by one to the previous amino acid at the C terminus of the growing chain. Each addition involves the participation of protein elongation factors and occurs in 3-step cycles as each amino acid is adding to the preceding one. Energy expenditure occurs in the first and the third steps. One more GTP is hydrolyzed to provide energy for this overall translocation step. The mRNA is moved through the ribosome in one direction, 5' end first, equivalent to the ribosome moving 5 to 3 on the mRNA. The ribosome and the mRNA move relative to each other, codon by codon. This elongation cycle takes less than 1/10 of a second in bacteria and is repeated as each amino acid is added to the chain. So the structures are here. The new tRNA will come in. There will be codon recognition. Energy will cause the binding to the proteins. That will then lead to peptide bond formation. Energy will then be used to translocate, moving the one that is now carrying the protein to the P site, and the previous one will leave through the E site. Overall, the end process is termination. Termination is the stage in which it is finished. It begins when a stop codon enters the ribosome, triggering a series of events that separate the chains from its tRNA and allowing it to drift out of the ribosome. A release factor protein will bind to the stop codon and cause hydrolysis. So it's going to cause water to bind to break the bond. The new peptides will then be released. The polypeptide is gone through the exit tunnel. The complex will then dissociate, and the parts then can be used to make a new structure. The ribosomal subunits can be used again. After termination, though, the polypeptide may still need to fold into the right 3D shape, undergoing processing, getting shipped to the right place, or combining with other polypeptides. So we are not done. This is just the termination process of translation. Overall, this process can be occurring simultaneously in different organisms in different ways. Which one of these is a bacteria should be fairly obvious because you have DNA and RNA in the same place on the right. That is the bacterial form. So the structures, the images are a little confusing, I agree. But polyribosomes, whatever, you can have multiple ribosomes attached to the same mRNA. You can have multiple RNA polymerases attached to the same DNA. This can happen multiple times, and it does speed up the process. So we can see that mRNA and RNA polymerases in the actual electron micrographs are basically barely visible. The proteins that are produced are even too small to be seen in these images. The ribosomes and the polyribosomes are large enough to be seen, but this structure is occurring. Overall, the folding process is typically involved through a combination of tertiary and quaternary structures, as well as chaperonin, like we talked about in previous units. But another thing that happens within this process is the targeting of the polypeptides. Almost all proteins that are destined to be secreted have a sequence consisting of between 5 and 30 hydrophobic amino acids on the nitrogen terminus, which is the signal polypeptide. It's a leader that tells the cell where to send the process. Free ribosomes suspended in the cytosol synthesize proteins that reside and function in the cytosol. Bound ribosomes make proteins that will be exported. So the first thing that's released is that signal polypeptide, and the signal recognition particle, the SRP, will match. And if it's something that's going to be excreted, that free ribosome will become a bound ribosome and will produce the protein directly into the endoplasmic reticulum. That signal is removed and is not part of the final protein, but it's the first thing that comes into its set. Other kinds of signal peptides are used to target polypeptides to the mitochondria, chloroplast, nucleus, and other places. In these cases, translation is completed in the cytosol before the polypeptide is imported into the organelle. And so while the mechanisms of translocation vary, each of these polypeptides is essentially a zip code that ensures the correct delivery. Bacteria even do this, even though they don't have an endoplasmic reticulum. They employ signal sequences to target proteins to the plasma membrane for secretion. So it is something that's found in all organisms. Overall, sort of the summative process for us, okay, RNA polymerase II will lead to the production of mRNA from DNA that will then be processed. You will add a cap, you will add a tail, you will splice the exons together. The introns will stay in the nucleus and the exons will be expressed or will exit the nucleus. Without the cap and the tail, the mRNA cannot leave the nucleus. The cap will allow it to bind to the small ribosomal subunit. The large ribosomal subunit will bind in connection, so the mRNA and the rRNA will come together. The tRNA will be activated and charged by the synthetases and will carry amino acids into the ribosome structure. That will then use GTP to attach these molecules together to build the growing polypeptide. That's pretty much it. Okay, if you look at sort of in the closer process, again, cap binds, large subunit comes in, protein grows through this process. Folding and modification will follow translation. The primary structure determines the secondary and tertiary, chaperonin. Post-translational modifications may occur, but that's all in future sections. Although bacteria and eukaryotes carry out transcription and translation in similar ways because they evolved from the same sources, they differ in the machinery and the details of the processes. Transcription is terminated differently. Polymerases differ significantly. Ribosomes are different. Okay, you just need to realize overall that most of the differences occur because of the complexity of eukaryotic cells. So in the absence of a nucleus, a bacterial cell can simultaneously transcribe and translate, and this process will be significantly faster, but will offer less points of control and more points of mutation and error. The additional steps in eukaryotic processing allows for coordination and the elaborate activities that a eukaryotic cell is capable of. I hope this made sense. If you have any questions, please talk to me.